Hello everyone, in this video we will discuss important difference from the chapter Biomolecules. These are very important questions which are frequently asked in the CBSE board exam. So first difference here, the fibrous protein and the globular. Fibrous is a thread-like structure, whereas globular is folded to form spheroidal shape. Fiber, because it is fibrous or fiber-like structure, it is insoluble in water, whereas globular is soluble in water. Now this is a 1 mark question. What type of a bonding hold them together? So fibrous is holded by the hydrogen bonding, whereas the globular involve all types of bonding which exist in proteins, that is disulfide bridging, intramolecular hydrogen bonding, van der Waals interaction, as well as dipolar interaction. And the important part here is, what are the example of fibrous protein? Keratin, collagen, fibrin, myosin, and in case of globular it is insulin and hemoglobin. Now the next important difference is glycosidic or peptide linkage. They can ask in a simple definition form also. What is a glycosidic linkage? The ether bond between two carbohydrates. So here it is OH and OH, there is loss of water. So due to which there is a ether bond, and this linkage is called as a glycosidic linkage. And the important point, some of the examples are stated like this. What is the hydrolysis product of sucrose? So it is glucose and fructose. Maltose, these three are the disaccharides, sucrose, maltose and lactose. So on hydrolysis they will give two carbohydrates. Now important thing or easy way trick to learn these hydrolysis product, in all the cases one of the constituent is glucose. In case of lactose, second one is galactose, sucrose, second one is fructose, and in maltose it is a 2 glucose unit. So from the glycosidic part you must know the definition, and then there is a glycosidic linkage between all these. Then what is a peptide linkage? So it is a part of a protein. So here two amino acids, or we can say amide bond linking two alpha amino acids. So these two are the alpha amino acids, where amide linkage is formed by removal of water, COH, OH from here, H from here. So this is CONH, amide linkage between two amino acids is known as a peptide linkage. Next important difference is between DNA and RNA. So in DNA a deoxyribose present, whereas in a RNA ribose sugar is present. The bases present here are thymine, cytosine, guanine, adenine bases are present. In this case instead of thymine only uracil, rest all are same. So uracil, cytosine, guanine, adenine are present in RNA. DNA is double helix, RNA is single stranded. DNA occur in the nucleus of the cell, whereas RNA mainly occur in the cytoplasm. DNA control the transmission of hereditary effect, whereas RNA control the protein synthesis. So all these 5 points are important, and this question can be framed in 2 to 3 marks. Then the next difference is the nucleoside and the nucleotide. Nucleoside, it contains two basic components of nucleic acid, a pentose sugar and the nitrogenous base. Base are all the same, that is cytosine, guanine, adenine, thymine, guanine, uracil. Whereas nucleotide contain three basic components of nucleic acid: pentose, nitrogenous base and phosphoric acid. So nucleoside में सिर्फ दो पार्ट प्रेजेंट है, और न्यूक्लियोटाइड T means phosphate or phosphoric acids are present. So these three are the important part. In nucleotide 3 components are there. In nucleoside only second 2 components are present. Now next difference between the reducing sugar and the non-reducing sugar. So sugar which possess free aldehyde or ketonic group is known as a reducing sugar, which do not possess free aldehyde or a ketonic is a non-reducing. Reduce Fehling as well as tolerance reagent. The reducing sugar will reduce these two, Fehling as well as tolerance, whereas non will not reduce any of these reagent. Now examples, all monosaccharides are reducing sugar, and maltose and lactose is also a reducing sugar, whereas all polysaccharides and sucrose is one of the disaccharides which is non-reducing. So these examples are very important. Sometimes MCQs are framed from this topic. Now the next difference is between amylose and amylopectin, and both are the component of starch. So amylose contain 15 to 20% of starch. AmopPtin is 80 to 85% of starch. It is soluble, whereas AmopTin is water insoluble. It is a simple, long, unbranched chain where linkage, glycosidic linkage is in between C1 and C4 carbon of two carbohydrates unit, whereas in AmopTin it is a branched. So in addition to C1 and C4, there is also a branching between C1 and C6 by this glycosidic linkage. So this question, in addition to difference, also can be framed in a form of short note. Next is regarding vitamin. Vitamins are of two types, fat soluble and water soluble. Examples A, D, E, K. Water soluble, vitamin C and B. These vitamins are absorbed more easily by the body in the presence of dietary fats, whereas these are not stored in the body and easily excreted in urine, so need to be taken regularly in diet. So this is a 1 mark reasoning question, that why vitamin C is not stored in our body, or why it is excreted out through urine, or which vitamin we have to take regular in diet, because vitamin C is not stored in our body, so we have to take regular in our diet. Next is essential amino acid and non-essential amino acids. So those amino acids which are not synthesized in our body and to be taken in diet is called as an essential amino acid. So these are not synthesized, so we need to take it in our diet. For example, valine, isoleucine and lysine. Non-essential, these amino acids are easily synthesized in our body, so there is no need to be taken in diet, and hence they are considered as a non-essential amino acid. For example, glycine and alanine.